So due to the physiological processes, there are some pathogens that they aren't compatible. They won't recognize us as a viable host, which is kind of nice. You need to have the correct chemical response receptors. Those aren't available always. It depends on what kind of virus it is. Bacterial phages recognize bacteria. They're not really going to recognize human cells. Also, pathogens need, they have needs for their environment as well. And so we might be incompatible for the pathogen's survival due to temperature, due to pH, there's a whole number of reasons. So for some, we have some resistance, but we don't have an innate resistance for some pathogens. Okay, so it's only some of them. So we have, structure, chemical, the processes. These are all processes that prevent pathogens from entering the body. We have a nice protective layer called our skin, our mucosal membranes. These are great at helping to prevent viruses. They kind of stick them in places so that way they can't, they either can't penetrate or they're stuck in the mucosal membrane. So, host defense. We have three basic lines. First line is our external physical barriers for infection. So that's the skin, the mucosal membranes. This is why, if you have a cut, is when you really need to be careful when dealing with some of the, infectious particles. So that's why it's always important to know. In a lab, the most dangerous is playing with broken glass and some kind of thing that could be infectious. Because if you spill some infectious particle on rubber gloves, no big deal. But, and you have the rubber gloves, you have the, your skin, you have multiple layers of infection. But if your gloves have a cut in them, and it cuts your skin, then all of a sudden you've just given that pathogen direct access to your blood. You bypass that first line of defense. Second line, internal, non-specific host defenses. So this kind of helps protect cells and bloodborne chemicals. So just kind of, we have our protective cells. Our blood has a bunch of things in it. It circulates through our body, tries to protect us. It has a lot of non-specific, just kind of random. We're trying to help the body. Third line, if that doesn't work, if it's gotten past our defenses, it's in the body. If the non-specific, just kind of general stuff doesn't kill it, then we start having to get to the specific immune system. And so, exactly, what do we need to specifically respond to this pathogen? How can we take this out? So, makes sense, those three different levels. Prevent it from coming in, otherwise just kind of try and have generic stuff. That doesn't work. Go hardcore and specific.
Where would the acid in your stomach fit into first line or second line?
Acid in your stomach? Because that's, the idea would be that when you get to your stomach, because it has to get through your mucosal membranes. So you even prevent that. And then once it's in your stomach, then maybe that would tear it up enough, so I guess be in the first line before it gets to your blood. So then once it's in your blood, you have to deal with the second line, which is more of the non-specific stuff.
So the bacteria being fried, the bacteria that make it as far as the stomach can get fried by the stomach acids, pH would be first line?
I don't think so. Because, that's kind of a physical barrier. But a lot of things get, it's more the mucosal membranes and skin that really provide that first line. That's why we have them in all of your air pathways and everything. That's why cystic fibrosis is so bad, because that mucosal membrane does not ever fluff out and leave, and so all those pathogens can just stay there. So then they can start to grow and double versus being taken care of and gotten rid of. Just to give you an example, this is our skin. If you actually look at the surface of the human skin way up close. Okay. So, skin is a physical barrier. There's chemical factors. It has low pH, salt, lysozyme. So it has things that help prevent and stop these microorganisms. There's also competition because we have that normal microbiota that we want. You know, we have microorganisms growing in us, on us, that are good. And so they outcompete, hopefully outcompete some of the infectious ones. And so it's that idea of as long as we can keep the good stuff there, then hopefully any bad stuff just kind of gets fluffed away. Epidermal dendritic cells, so, you know, there's some that specialize. You have non-specific phagocytosis. They'll just kind of, we have specific cells that will go through, they kind of hover around the mucosal membranes, and they randomly sample things and go, ooh, this is bad, we should figure out how to kill this. And it's kind of randomly do non-specific phagocytosine. So what is phagocytosine? Phagocytosis?
Bring it
Going on there,
Eats the
grabbing it.
Yeah. And is it solid or liquid?
solid.
Okay, what is liquid?
Pinocytosis.
See, the vocab's important. So, phagocytosis randomly grabs things, kind of starts chewing them up. Also, then that way it can, you know, if it detects something, it can either just destroy it, it can also send maybe pieces off to be like, sort of recognize this is bad, and so kind of alert the rest of the body. We have our mucosal membranes. They trap pathogens. That's why we get boogers, because, you know, all that dust and debris in our nose gets trapped versus getting to go into our body. Okay? We have ciliated cells. So this is for our respiratory system. So as we breathe, we have mucus, but it also has cilia. So that cilia can come along, kind of brush that stuff out. And so keeps it moving. So that way you don't end up having it sitting there. We want to clear that mucus. So you keep having fresh mucus come on, mucus traps it, and then you want to get rid of it because you don't want it just to sit there with pathogens. We have lysozymes in the nasal mucus. The parts of our body that are going to get a lot of these microbes. We don't just want to trap it. We kind of want to also have things there that will start to destroy them. So that way, you know, if you're only getting a couple, you can trap it and move it away. But if you're getting a barrage of things, you kind of want to trap, start destroying, so that way if any try to sneak through, you've already weakened them. That's what lysozymes are good for. Other first-line defenses, sorry, I wrote this a while ago or made this again. So stomach acid, first line. Lacrimal glands, so tears, removing pathogens. They also contain lysozymes. So it's one of those things.
I'm sorry, I never knew that. Tears contain lysozymes.
There's reasons they don't taste good, besides the fact that you're sad. But yeah, those are other things that just kind of, any way that things can enter our body, we try to have some form of defense. Okay. Our skin is composed of two basic layers. We have the epidermis and dermis. Okay, and so that epidermis, which is what I was showing you earlier, it's very, very tightly packed. So you aren't going to really get things packed past those cells. There's very few pathogens that can actually get through our epidermis, the outside layer of our skin. And we're constantly shedding it. That's why we have dead skin cells. That's why it also looks so flaky. But there is epidermal dendritic cells, which will do that phagocytosis. And then you have the dermis, which is where you have the collagen fibers, and they help, you know, resist abrasions. It's what gives our skin a nice elasticity. And so this is also where all the hair follicles, the blood vessels, the nerve endings, all of this is in the dermis right underneath that epidermal layer of protection. And the sweat glands will start to secrete not only perspiration to cool us off, but they secrete the salts, the antimicrobial peptides, the lysozymes. And so lysozyme is just an enzyme that will destroy the cell wall of bacteria. So we start, so it's, you know, in our sweat, we start secreting things to kind of try and protect us. Okay. Salt inhibits pathogenic growth. Antimicrobial peptides also kind of inhibit microbial growth. And then you have lysozymes destroy the cell walls. We mucosal membranes. They line all body cavities open to the environment. So let's see if I can give you other pictures of these. So you start thinking about things that, line, this is our respiratory system, so you have to have this, you have to have protection because we need to breathe, which means we need to take in microbes because they just inherently are in the air. It lines the lumens and of the respiratory tract, and so it just lines everything so that way, your respiratory tract, your urinary, gastrointestinal, and reproductive tracts are all lined with mucosal membranes to try and protect us. So, just kind of a visual of where all of these are, where they are in our actual body. And then what is the cellular defense? We start moving on to that secondary level. Plasma, so liquid portion of blood. It contains complement proteins, and hemoglobins. It has our host defense system is in the liquid part of our blood. It's mostly water, but it contains, electrolytes, dissolved gases, nutrients, proteins. So, it contains everything that we need to keep taking around our body and help our body out. But it also has some basic things, that can just kind of help as a line of defense. Okay. So we have formed elements, you have cells, you have cell fragments, erythrocytes, red blood cells, leukocytes, white blood cells, platelets, cell fragments. We'll talk about all of these a little more. So the platelets, these are involved in blood clotting. So you get a cut, I don't want those platelets to come and start clogging that so you don't bleed everywhere. Red blood cells carrying iron, white blood cells your immune system. Okay. And leukocytes is a very big general term. When we're talking about leukocytes, there are multiple classes of leukocytes with different types of jobs. A lot of them are involved in phagocytosis because you just want to engulf whatever it is, chew it up, get rid of it, make it go away. Some of them actually, though, will cause inflammation. They'll cause some kind of allergic response. If you're talking about B and T lymphocytes, then you have antibody synthesis. So this is where you get your cell-mediated immunity, because this is where they actually figure out how do we kill this, how do we make an antibody, and then you have lymphocytes that make the antibody, and you have lymphocytes that remember how to make that antibody. So if you ever come across that pathogen again, it's like, wait, we have this, we know what to do. Okay? So macrophages, monocytes, things that just they start phagocytosing whatever kind of parasites or microorganisms.
Is there any major difference in the action of neutrophils and monocytes and macrophages in the way they act in phagocytosis? Or are they basically just doing the same job?
I think they recognize different things, which is why you need multiple things. So, they're all part of that general immune system that's trying to just help us. Let's see, do I write out exactly what they all do? Neutrophils, so they're all part of that. So you can see they look different and they have different things they do. So if you're talking about different ones that are capable of the.
Those look familiar.
But this is the inflammation, causes the inflammation, and the allergic response, the leukocytes, or the lymphocytes, which this is where we're going to get our B cells and T cells. So this is our immune system, our antibody synthesis. This is, as you can see, just kind of does the phagocytosis, defends against parasites. Just kind of looks a little different than over here. Neutrophil just does phagocytosis. So just kind of some of the differences in the cells. They do different things. They grab different things. They recognize, and and also just kind of a what is the percentage of blood. You know, ideally, we don't have a lot of the parasites that we need to kill, so we don't have a very high number. Whereas we have a lot of things that just need to be kind of engulfed and dealt with, so we have a much higher number or percentage of the neutrophils. Okay. Broad category of our host defense, white blood cells, keeping it kind of surface level. Okay.
Why are the ones called granulocytes and the other ones agranulocytes? Is it the texture of them?
The granulocytes, they contain large granules that stain different colors. There's the 3 basic types, as you can see listed. So, like they stain blue with basic methyl blue dye, or they stain red and orange with an acidic dye, or they stain lilac with a mix of the acidic and the basic dye. So, how they stain, you can tell. And then the neutrophils and so they're part of the phagocytosing of the pathogen. So just kind of like, okay, so these stain that way. The antigranulocytes, the cytoplasm appears uniform under light microscope, so you don't see that rough texture.
So up to 70% of all leukocytes are neutrophils?
Kind of, approximately. I mean, 60 to 70, because you also have 20 to 25% or lymphocytes. Kind of want, I mean, these are the 2 major things. You either want microorganisms to be destroyed, or you want to have a way of making antibodies to destroy these things, recognize them. Okay. Monocytes, they leave the blood and mature into macrophages. And lymphocytes, most involved in adaptive immunity, so the actual specific immunity. So they all come from the same basic stem cell. So blood stem cell and bone marrow. So our bone marrow is what makes it, what causes issues if you have problems in your bone marrow. The hepatocytes, so development of stem cells in bone marrow to form elements. So we keep trying to form, generally our leukocytes, our erythrocytes, platelets, things that are going to, are they going to give us the innate immune system, so the second line of defense, adaptive immunity, so the difference between just general immunity, going to kill everything, versus very specific immunity. Remember we talked about those 3 lines. Prevent it, basic kill, specific kill. Okay? Does that make sense? Okay, so basic stem cell can slightly differentiate into these 3, saying, okay, it's still the blood stem cell, so it's going to go into the blood pathway of differentiation. Makes one decision, goes into one of these 3, and then from there, it can either go, lymphocyte. You're going here, you're going to become a lymphocyte. But if you become a myeloid stem cell, then you have some choices between what you become based on what's needed. And you can see the difference of the granule because of the way they're textured. Or you can decide to lose your nucleus and become a red blood cell and transport gas. Okay? It's making more sense.
Specific, non-specific, can you go over that part again?
Oh. So innate versus adaptive. So innate immunity, that second line of defense, because they're going to go around just phagocytosis, just eat whatever's there. Versus if you're doing adaptive immunity, this is your antibodies. So that's very specific to a certain thing. Whatever's causing your immune response, and you make antibodies, that's adaptive. So that's very specific. Medically speaking, the lab will analyze your leukocytes, so general class of leukocytes. And so your differential white blood cell count, they can signal diseases. I mean, if you don't have the white blood cells you should in your body, then obviously something's wrong. If you have elevated white blood cells, then your body's trying to fight something and has produced more of them. So it's one of those things by, if you're, increased, you have some kind of allergy or parasitic worm or some kind of infection. Decreased, you might have something like HIV or something that's actually decreasing your white blood cells. Either way, it's not good, but it's a good sign of something's wrong. We need to look into this. The bacterial diseases also show increased leukocytes and neutrophils. And viral infections show increased lymphocytes. So specific trying to. So it's one of those things, we're looking at what's going on in the blood, tells us what's going on. So lymphocytes are a lot of those natural killers. They secrete toxins on the surface of virally infected cells and tumors. And so they differentiate from normal body cells because they have membrane proteins similar to NK cells. So the natural killer lymphocytes especially go there, and their specific job is just to kill things. Phagocytosis, we keep talking about this. Steps, you have chemotaxis, chemical attraction. So there has to be some way for our natural immune system to say, this is wrong, we need to eat it and destroy it. And so it has to have some kind of chemical signal, some chemical attraction. Then you need to have adherence, so attached to this.
Is the chemotaxis in any way different from the antigens which normally attract antibodies?
Antigens trigger an immune response, because an antigen is anything that triggers an immune response. And so those are still going, I mean, they're going to be giving some kind of signal somehow.
Are antigens just much more specific than chemotaxis?
They're just different kinds of signals. I mean, chemotaxis, it says this cell has some kind of signal. It's producing some kind of signal saying, I'm not right. Versus an antigen is anything your body recognizes as foreign. So that can be a protein.
I thought an antigen fit into one specific antibody, or an antibody fit into one specific antigen.
You can make a specific antibody for the antigen, but in general, antigens can be multiple things, whatever messes with your immune system, whatever triggers an immune response. Right? But in general, if we're talking about phagocytosis and we're trying to eat the bad cells, you want some kind of way of recognizing it, so chemotaxis. So you move either cells towards or away, chemical stimuli. So they end up attracting phagocytosing leukocytes to site of damage or invasions. So you get a cut, a lot of times they'll send a signal that says, hey, why don't you guys come over here and check this cut out and make sure that nothing got in while we have this breach. You want to attach a phagocyte to the microorganism, so whatever's there, you want to attach, you need to have attachment, adherence, ingestions. You start engulfing it, so eat the whole thing. And then killing, digestion. You have that breakdown, so it gets broken down, you fuse, you add some lysosomes, just chew the thing up, get the parts, and then you can just exocytose it. Any undigested remnants, you can just kind of get rid of everything. So, picture-wise, okay. So chemotaxis, some kind of message saying, come over here. Adherence, you start to attach, and you start seeing the little pieces, the pseudopods, move by chemotaxis towards whatever microorganism they're trying to get rid of. So you get that adherence. Ingestion, bring it in. By phagocytosis, start making a little space so that you can break it. You also signal to the Golgi going, make us some lysosomes. Okay, good. So that is can come along. You fuse them together. So the fuse of a series of vesicles. So basically, you make sure you fuse digestive parts with your microorganism. You have this vesicle. You go, this has something toxic in it. I want it to be chewed up. Make sure you bring lysosome over, chew it up. you have the residual body parts, you know, you can take whatever's useful from it, get rid of the rest of it, and just kind of eliminate exocytosis. Okay. Make sense? We've kind of talked about it before, basic differences. Instead of some random food source, it's particularly specifying on microorganisms in our blood. Biology repeats itself. A lot of things it works, so why change it? So you see the same processes over and over again, with maybe slight tweaks and modifications for that specific use, but the general gist of it stays the same. Complement cascade. So, set of serum proteins, they're important for directing a rapid host defense. So we have a classic, alternative, and lectin pathways. And so we're trying to figure out how we're going to phagocytose these. Membrane attack is complex, and so how does the cell lyse, or how do we prevent the invading microbes? And also we end up getting a lot of times the inflammation. I think movie might make more sense. Let's see how well these
Complement system is a set of proteins that circulate in the blood serum. When activated, these proteins work together to destroy foreign cells by cytolysis, to activate the inflammatory response, and to assist in phagocytosis. The main components of the complement system are called C1 through C9.
And for activation, we're talking about the classic pathways.
Complement can be activated through three different pathways: the classical pathway, the alternative pathway, and the lectin pathway. The classical pathway was the first activation pathway of the complement system to be discovered. The classical pathway begins when a pair of antibodies attach to antigens, such as proteins, or a large polysaccharide on the surface of a bacterium. The antibodies bind and activate C1. C1 then cleaves several copies of C2 and C4. Together, the C2A and C4B fragments form an enzyme that cleaves C3, activating the complement system.
They're incredibly complicated, as you can see. I see that. Versus,
What do we need to know about that?
so the whole point is to get that cellular response here. We're trying to.
The alternative pathway of complement activation was so named because it was discovered after the classical pathway. While the classical pathway requires antibodies specific to the invading microorganism, the alternative pathway requires three complement proteins called factor D, and factor P, or propertin. These proteins are attracted to microbial cell surface material, such as the lipid carbohydrate complexes of certain bacteria and fungi. Together with C3, these proteins result in the cleavage and activation of C3 proteins, which in turn activate the rest of the complement system. The most recently discovered pathway of complement activation is the lectin pathway. When macrophages ingest material through phagocytosis, they release chemicals that stimulate the production of carbohydrate-binding proteins called lectins. The lectins bind to distinctive patterns of carbohydrates on the surfaces of certain bacteria and viruses. The lectins themselves can act as opsonins for phagocytosis, but also activate C2 and C4.
Okay, so just kind of to give you some general look of what we're talking about, because, I mean, it's complicated. There's multiple, you know, there's the classic approach, there's the alternate approach, there's the, you know, the entire point is how are we getting the message, you know? Classical approach, we have antibodies that are attacking it, so our body can recognize there's an antibody, that means we need to go kill something. Or do we already have some basic generic things for the alternative pathway? The B, the D, the P, going, okay, so we have these proteins, they recognize generic microorganisms. They go, find it, they find their happy spot to bind, and then they will start the cascade. You know, or we have lignans, so it's like, well, we know a lot of them have this, can we, so it's all different ways of trying to basically cascade down to that membrane attack, that complex. Okay, so we're trying to just figure out different, you know, our body has a lot of different ways of activating, saying, we're under attack, come help and destroy this. That part makes sense? Okay. So, this makes it a little simpler, a little nice diagram with the, you either have an antigen is whatever is foreign, your body recognizes as foreign. It doesn't have to be a chemical signal, protein, sugar, carbohydrate, whatever. Whatever your body recognizes as foreign, entire microorganism. If you already have antibodies against it, then you start going through the classic pathway, activation, your C3, C3A, you know, basically you cleave, you have a bunch of these things floating around. When they get cleaved, then they start becoming active. Okay? So you can, some of them, you know, when you start getting those C5s get cleaved, you start getting inflammation. And so heating things up helps to kill and destroy protein. So, you know, part of our immune system is that inflammation. So either you have antibodies, you have basic factors that recognize the foreign things, or you have those leptins that will recognize specific sugars that we don't necessarily have on our cells, but that are classic on microorganisms. Okay. And all to go for a membrane attack complex and cell lysis. The entire point is, that's foreign, let's go attack it and kill it. Okay. So, classic, antibodies bound to microbes, alternative, initiated by microbial cell polysaccharides, lectins, bind to the fungi, bacteria, and viruses. Okay. All pathways meet at the C3 protein, which is then split. Does kind of make sense? Make it so that that complex pathway looks a little not so complex. It's all the same idea. Okay. So our outcome. I don't have that. There we go. Okay. So the outcomes of this, you have microbes that end up becoming coated by antibodies and the C3b proteins. So, you know, it's one of those things where you start getting coated with antibodies, that just is even more of a basically neon sign to the macrophages saying, eat here, come here, engulf this, this is wrong, we're trying to slow it down. Okay, so that's why it enhances that phagocytosis, is because they're huge big signals with those antibodies, because antibodies just start attaching, and you just get bigger and bigger signal. Inflammation stimulates the chemotaxis of leukocytes. So you get that heat rising, you start going, something's wrong here, we need to start targeting things here, start, you know, bringing help to deal with whatever is causing this issue. So it's one of those signals. And so it's like, you know, when you get little cuts or you start feeling that inflammation, it's because your body's trying to fight whatever's there. So this is where we get the C3A, C4A, the A's. You also get membrane attack complex, the MAC. Okay. So cytolysis, this is where you get the proteins from the lethal, form lethal holes in the microbial membrane. You start punching holes in it, nothing really lives very well with holes in its membrane. This involves the C5b, the C6, the C7, the C8, C9, feed complement fixation. And, you know, you're trying to basically get to that inactivation. So once everything has come, you've killed whatever you need, you punch your holes, you then want the cell to turn down, you shut off. So it occurs quickly, you break down of the activated components, so then you can make your immune response, go, kill whatever you need to, and then stop. So that doesn't go out of control and start telling cells we need. So this is where they're showing kind of class pathway, complement pathway. So you have your antibody, C1, an active enzyme when it binds to the antibody antigen, it's cleaves other enzymes, and so you start getting this cleavage. The fragments come together, they bind and form this third component, which goes and causes chemotaxis, phagocytosis, and inflammation. So you get, okay, they send signals, which also comes up here, we start making our enzymes, so that you can make these, these messages, and then go along, cause chemotaxis, the phagocytosis, the inflammation, and you can see you start making these holes in the actual membrane of the pathogen. So all these little pieces, this cascade down, is just so that you can start chopping up and getting and going, okay, here, start putting these here, and then water can rush in, and cells can't really handle water because what? What will happen if you start letting a lot of water in? They'll swell, they'll lyse, they'll burst. Okay? So you end up getting, you need all of these little chemicals or all these little signals come together with the C9 so that you can form that membrane attack complex. So this is the membrane attack complex. Okay, so it's attacking the membrane, hence the postological name. And it drills a circular hole for pathogen cytoplasmic membrane, so it leads to the lysis of the cell. Okay. This kind of makes sense. We're starting to see this pattern. Things get activated, they get cleaved, so they're activated, and they go join up with other partners and other halves, so that they can go cleave other things and form complexes to basically put holes in membranes and kill pathogens. Okay. So this is an actual picture of what a membrane attack complex looks like. So, that's pathogen, and all of those are membrane attack complexes. That's a lot of holes in that membrane. Pathogen's not going to do well for very long.
Just a couple holes wouldn't do the job.
Oh.
Excessive.
I'd rather it go excessive on a pathogen than decide that, well, we're all activated, I'm going to go find some other cell to do this too. So, it probably just really helps to make sure it doesn't give it time. Because, some holes, still get the water in, but if you really want, you want it dead now, not dead in an hour. Interferon. Type 1, we have alpha and beta interferon. They're produced by virus-infected cells. And so what they do is they stimulate cells to make antiviral proteins. So it's kind of nice. We have ways of dealing with the viruses. So the ABPs. And type 2 gamma interferon, so alpha, beta, gamma. In case you didn't know, those are the Greek symbols for an alpha, for a beta, and for a gamma. So the gamma interferons, they're produced by activated the T cells and then those natural killer leukocytes, the NK cells, okay? And so these come through and they stimulate macrophage activity. And so they're going, this cell is infected, kill this cell. Okay? So these are used for like hepatitis, multiple sclerosis. You can have that chronic granulomatous disease. So interferons are used for multiple things. So these are non-specific. They inhibit the spread of viral infections. And then
I will stop here.
I think we'll leave keep going. Inflammation.
Oh.
We have a little bit of time. The more we get through, the less will be later. Okay. sorry, alpha and beta interferons. So alpha and beta are type 1 or type 2?
Type 1.
Type 1, okay? So, we have a virus. We have double-stranded RNA. So, in this case, the cell recognizes it. You have an interferon gene. Goes, we're infected. And so, this tries to then, viral replication in the cell triggers the transcription of these interferons, the alpha and the beta, and it depends on the type of the host, but basically, that transcription turning on of that viral gene sends a signal. So the interferons start, leaking out of the cell going, there's bad stuff going on in the cell now, kind of being taken over. So that interferon release diffuses to neighboring uninfected cells, binds to their receptors, and then that binding triggers transcription and translation and activates the ADP genes, okay? ADPs. So what are ADPs? The antiviral proteins. So this cell's infected, sends a signal going, help, I'm infected. The cell then, seeing that, getting that help signal, triggers proteins that are antiviral proteins, so that as time passes, you start getting a buildup. You have the inactive ones, but then they become active when they attach to the double-stranded viral RNA. Double-stranded RNA is not necessarily a good thing. We don't have long stretches of double-stranded RNA in general. That gets chopped up. But to have these antiviral proteins, they'll kind of recognize this double-stranded RNA. This is not good. Let's attach to it. Let's take it. And so it gets degraded, and so it binds to the ribosome, which stops the protein synthesis and viral replication. So it's a way of actually going, this is double-stranded RNA. This is not good. Let's bind it up, gum up the works, stop all production, because we don't want to make this virus anymore. So we will sacrifice our own ribosomes and make it so they can't work just to stop this from being replicated and translated. Okay? So this makes it so that this neighbor cell is protected at a later time. So it doesn't protect the host, but it's the host kind of last dying effort sends out a signal going, you guys protect yourself because this is coming. Okay? So the infected cell dies, releases the virus. So it's one of those things where it's a response you can't have in a single-celled organism, because if a single-cell organism dies, it's dead. But in a multicellular organism, a lot of things happen where we have the death of a cell because it's good for the whole. This is why we have programmed cell death. That's why we have apoptosis, because that single cell dying is good for the whole. Okay. So, the cell that's infected, it's done for, but it can warn all of its neighbors so that they can fight off the infection.
So interferon is sort of like an early warning system for future cells which might become infected?
Inflammation. So there are 4 cardinal things: redness, edema, pain, localized heat. Okay. Rubor, swelling, dolor, calor. So why do we have inflammation?